Is it okay if I do the introduction? Okay, right. Yeah, no problem. Thank you. All right, thank you all so much for coming today. So I'd just like to start with a quick land acknowledgement. We would like to respectfully acknowledge the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We also recognize the Inuit of Nunatsiavut and Nunatkavut and the Inuit of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all the people of this province as we work towards true reconciliation and honor this beautiful land together. So this week we have Dr. Lindsay Cahill. Lindsay did her PhD in chemistry at McMaster University and completed a postdoc fellowship at the University of Warwick in the UK. She then joined the Hospital for Sick Children as a postdoctoral fellow and research associate using biomedical imaging to study mouse models of human diseases. Dr. Cahill joined the Department of Chemistry and Radiology at Memorial University of Newfoundland in 2020 and is currently an associate professor. Whenever you're ready.
Thank you very much. Thank you for the introduction and thank you to everyone for being here. Keep track of my time. All right. So this just summarizes my bio in a slide. And I think my research program in chemistry is combining all of these different aspects of my training. So using NMR during my PhD at McMaster to study lithium-ion batteries. Then I used magnetic resonance in more imaging related applications when I was at SickKids studying mouse models of human disease. This is a recent picture of my group. And one of the themes of our research is studying environmental exposures and their impact on human health. We have a clinical component to our work, which I'll start off by telling you about, and then I'll get into some of our studies using mice. Today, I'm going to talk about two different environmental exposures that we're very concerned about, so microplastics and nanoplastics, and then I'll also tell you about some work we've been doing on perfluoroalkyl substances. So I'll start off with plastics, the good. Plastics, of course, we find them everywhere in our indoor and outdoor environments because they have a lot of favorable properties, right? They're very lightweight, durable, inexpensive, so they're quite versatile, and we find them all over. Plastics, the bad, we produce over 400 megatons of plastics annually. I'll ask you to think about how much of that plastic you think is landfilled or left in the environment. These multiple choice questions are not particularly hard. Unfortunately, the answer is D. So the majority of the plastics that we use ends up in the landfill or in the environment. And we've known for decades, I'm sure speaking to biologists, you know this better than I do, about the impact of plastics on the environment. These are just some of those shocking photos that you can find on the internet. Has anyone read Holly Hogan's book, Message in a Bottle? I highly recommend it to people. For those who don't know Holly Hogan, she's a seabird biologist. She's a Newfoundlander. And this book really talks about her adventures and her travels as a seabird biologist, but also places where she's found plastics where you wouldn't expect. So I really recommend that you read this book. I'm interested in what happens when those macroplastics degrade and form smaller plastics. So because of UV radiation, mechanical degradation, biodegradation, microplastics break down and form, plastics break down to form microplastics that are defined as less than a diameter of 5 millimeters to 1 micron, or even smaller into the nanoplastic region. There are lots of routes of exposure to plastics, so inhalation, ingestion, or through dermal contact. And there are lots of, well, I'll ask this question. So where in the body do we think microplastics have been found? You probably know when there's an all of the above. Usually that's the answer that you want to choose. And so starting in 2021, sorry, the slides are out of order. I wanted to say there's lots of sources of exposure. So dust, food, tap water, drinking water, cosmetics, clothing, microplastics have been found in all different sources of exposure, and then we've found them in all different body parts. So this is just a summary starting in 2021, different organs in the human where microplastics have been discovered, including this very recent paper that came out in 2025 reporting very high plastic levels in the human brain from autopsied samples. And we'll talk a bit more about that later. My organ of interest, though, is not on that previous slide. And so I'm interested in studying the placenta. So I'll tell you a bit about the placenta. It is, of course, a requirement for all successful mammalian pregnancies. It's a transient vascular organ. So you have a maternal and a fetal side, and it's the primary site of gas and nutrient exchange. So it's how the baby gets the oxygen and nutrients it needs, and then how the waste is removed. When placental development goes wrong, this can lead to a variety of pregnancy complications. And this can range from maternal hypertension to preterm birth, growth restriction, and in, unfortunately, the worst cases, stillbirth. So placenta development is very, very important, and it's important to be able to study when things go wrong and what causes this to happen. In 2021, an Italian group published the first evidence of microplastics in the human placenta. So they used Raman microspectroscopy. So they were simply looking at the sort of fingerprint of what the plastics were. So they would find a plastic particle and then determine what type of plastic it was. And they were reporting 5 to 10 micron plastics, mostly polypropylene, in healthy pregnancies. Important to note that these techniques are really just, are there plastics or are there not? It's not really giving you a sense of concentration or how much plastic is in the tissue. This resulted in a lot of media, lots of concerns about what is the plastic doing inside of the placenta and how is it impacting development and how is it impacting the pregnancy. I would say there's still a lot of media concern around microplastics. It resulted in a lot of different groups reproducing these results. So several groups have reported microplastics in the human placenta around the world. Microplastics have also been found in the meconium, so that's the baby's first waste. So that's telling us that plastics are coming from the mom through the placenta and crossing into the baby. And of course, babies are exposed to, not of course, but plastics have also been found in breast milk and in formula. So they're continuing to be exposed early in life. So my group is interested in looking at 2 areas that we consider knowledge gaps. The first is really what is the concentration of micro and nanoplastics that we're exposed to, and then what are the impacts of these exposures on human health. Working with several collaborators, we are interested in taking an exposomics approach to this, right? So we're trying to look at the totality of exposure. My group also does a lot of physiological monitoring, and we've been doing some metabolomics work to understand the impact on metabolism. Here is a picture of some of the group that's been working on this. Just down the hall is this mass spectrometry unit that we use. It's called the pyrolysis gas chromatography cyclic ion mobility mass spectrometer. And what's special about it is that it allows us to do quantitative analysis, which is different from the Raman or IR techniques that other groups are using. This is a picture of the pyrolysis unit. So we take these very small samples. I'm going to tell you about some of our work in the indoor air environment and in the human tissue samples. We take the small amount of plastic, heat it up to very high levels. And this is a 2D plot that shows us the decomposition of some of the common plastic types. So we're looking at polypropylene, polyethylene, polymethylmethacrylate, and polystyrene. And they each have their own distinct signature. We can integrate those peaks and then figure out how much we have. We're currently very actively recruiting through funding from Health Canada. So we have a site here in St. John's, and then we also have a site in Toronto. And we're really excited to compare Toronto to St. John's in terms of microplastic levels. We recruit fairly early in pregnancy, and we want to collect air and water samples from the homes. And then we take a maternal blood sample at around 28 weeks. And then we've been collecting placental tissue samples after delivery. So to compare the microplastic levels in the blood to what they're exposed to, and then also what we see in the placenta after delivery. So this is some very preliminary work from a student who was measuring plastics in the indoor air. She uses an active sampler, so she's pulling in air for around 24 to 48 hours. And it's size segregated. And she was looking at, this is the results for looking at particulate matter less than 2.5 microns. And you can see there's a lot of variability amongst these different sites that I'm showing here. And the majority, so in terms of plastics, the percentage is ranging from 9 to 65% of the plastics that we see, of the matter that we see consists of plastic. And most of that seems to be polystyrene, which we were a bit surprised at first, but then polystyrene is not very dense. So it's probably from all of your Amazon packages or the things in the home that it's floating around in the environment. So it may be overrepresented in terms of what we're pulling in through the sampler. So this is a very ongoing study. There's a huge debate in the literature in terms of how much plastic we're exposed to. Unfortunately, this term that humans eat 1 credit card of plastic per week has really made its way into the public space. I did an interview recently where they were talking about the paper where they found plastics in the brain. And they specifically asked me, do you think we eat 1 credit card worth of plastic? And I said, there's no evidence to support that. And then that was the title of the article in the Toronto Star, was related to eating a credit card's worth of plastic. So it's something that I'm really trying actively to fight against. There isn't evidence to support this. The paper that came out to suggest this was based on some modeling data that has since been shown not to support that idea at all. So it's still very unknown, but as I said, I'm trying to fight against this. There's a paper that came out at the same time in 2021 that suggested that we consume much less, so 4.1 micrograms per week, so 1 million times less than the 5 grams per week. Based on our results in the air, which again, very preliminary, suggests that we would consume around 160 micrograms per day. Now that would just be from breathing in the air. So that would take 85 years if we were gonna inhale a credit card's worth of plastic. Of course, there are other sources and there's still a lot of work to do there, but hopefully I've made my point about the credit card. I'm just gonna walk through some of the processing that we do for the placental tissue samples that we've collected. So far, 46 pregnant people here at Eastern Health have consented to give us their placenta, a small bit of their placenta after delivery. We try our very best to use what's called plastic-free protocol. So all of our tools are metal, all of our samples go into glass. But of course, all of that polystyrene that's floating around in the air can still end up on top of our samples. So we always collect procedural blanks with each sample that we collect. And there is a lot of background. So we background subtract out what happens with the procedural blanks. We do a lot of our processing in a clean room, which we're very lucky to have up in the 3rd floor in the chemistry space, again, to try and minimize some of that background contamination. So the tissue is digested with a strong base. We filter it off, and then we basically just take the filter paper and put it into the mass spec unit. So I'm showing here the results for this preliminary data set of 46 pregnant people from St. John's. Each of these 2-letter codes corresponds to an individual, and we collected 2 samples from every placenta. On the y-axis is the concentration in micrograms per gram, and the colors are the different types of plastic. There's a whole bunch of individuals where we didn't detect any plastics. You have to remember that that just means they're below detection limits, which can range from around 0.1 microgram per gram up to 1 microgram per gram. But those are considered non-detect. So there's a lot of participants where we weren't able to see any plastics. And then there's quite a bit of variability even within samples. Perhaps not surprising because the placenta is very heterogeneous. And a couple cases where we see higher levels, but I'll compare it to some of the literature in just a second. This is a study that came out at the end of 2024 from a group in New Mexico. That's the same group that's reported on the plastics in the brain. And you'll see the scale here, still in micrograms per gram, but it goes all the way up to 800. They reported plastics in every one of their 62 pregnancies. You can see the majority is this orange color, which is polyethylene. I will point out that the tubes that they collect their samples in are polyethylene, which is a criticism of this work. So we don't know what's going on. Their levels are very, very high compared to ours. They use the same pyrolysis mass spectrometry technique to measure their concentrations. Are there geographic differences? Likely yes, between New Mexico and St. John's. This is why we haven't started the study analyzing the data from Toronto yet, but it'll be really interesting to compare. I think there are some methodological concerns with their work in terms of contamination. So it's something that we're trying very hard to answer. We're working with a group in Amsterdam who also uses the same technique. And two of my students, Nikita and Justine, went there in November to run some of these samples in Amsterdam to compare the results, to try to get a sense of an interlab comparison. And we're trying to get more groups on board, which is proving to be quite challenging. But yes, there's an urgent need, I think, for us to really understand what these differences are so that we can move forward in terms of understanding the levels that we're exposed to. Out of that 46 pregnancies, so I'll say this is very early on, a very small sample size, we did want to look at whether there was any correlations with adverse pregnancy outcomes. So we did have a group within that 46. There were some pregnancies that had complications. So this included growth restriction, preterm birth, preeclampsia, and gestational diabetes. And so when we separated these pregnancies into complicated pregnancies and normal pregnancies, I'll note that this is a long axis on the Y. We did see that there was elevated levels in the male fetuses that were exposed to microplastics, had a higher level of microplastics. This is very consistent with what we see in the literature related to environmental exposures. Males often have more complications as a result of toxicant exposure. And there's a lot of speculation about why that could be. So this is something that we hope when we have our much larger data set, we're aiming for 300 pregnancies, hopefully overall, that we'll be able to dig into this type of data a bit further. Okay, so I'm going to talk a bit more about how we're trying to investigate the impact of plastics on human health. So one of the ways that my group does this is through the use of animal models of pregnancy. So we use mice, just located over at the Animal Resource Center across the way. There are many benefits to using mice to study pregnancy. So one is that the placenta in the mouse and in the human have a very similar vascular and cellular structure. Here is a micro-CT image of a mouse placenta compared to a piece of a human placenta. You can see the umbilical artery, the umbilical cord coming in and then branching into this really beautiful fractal pattern. So there is quite a lot of similarity between the two. Of course, the human placenta is much larger, and there are some other major differences that we have to consider when doing this work. Animals are, of course, very efficient for research, so low cost. They have very rapid gestation, so mice are usually 18.5 days. Very large litter sizes. The mouse model that we use can have up to 20 pups, so you can get a lot of placentas and a lot of fetal data from just one mouse, and they have very reproducible pathology. So we raise the mice in the same environment, they have the same exposures, the same diet, so we can really try to get close to causal relationships between the environmental exposure and the outcome that we're looking at. Humans are complicated. So this was one of our first study designs to look at the exposure of microplastics and their impact on pregnancy. We set the mice up to breed, and then they would get exposed to plastics through their drinking water. So we have a control group. Here we have 3 different concentrations of polystyrene microplastics at 5 micron in size, and then 3 concentrations of polystyrene nanoplastics, which were 50 nanometers. 1 day before birth, so at embryonic day 17 and a half, we euthanized the dam, and we weighed the fetuses and the placentas, and we measured the umbilical cord lengths. And here's a summary of these results. This was my master's student Zara's project. And here I'm showing fetal weights on the y-axis. This would be the control data that didn't get exposure to plastics. Red is the microplastic data, and blue is the nanoplastic data. And you can see as you increase the concentration, you see a decrease in fetal weight. And this was even more pronounced for the nanoplastic exposure group. So this was very concerning. Of course, we're seeing a relationship with fetal growth, suggesting that very high levels of plastics could cause fetal growth restriction in humans. We also saw this really interesting decrease in the umbilical cord length. And to us, the umbilical cord length is very telling. It's thought to be related to brain development. So as the baby's brain develops, it starts to move more, and as a result, the umbilical cord length grows over gestation. So if the umbilical cord length is not growing, it could indicate that there's something wrong with brain development. And that sort of motivated some of our subsequent studies that I'll tell you about. We also have a high-frequency ultrasound system over in the Animal Resource Center. This is very similar to what you would do in humans in terms of following fetal development. It just has a very high-frequency transducer, which allows us to get very high-resolution images. So this is an image of a mid-gestation mouse fetus, and we can measure the walls of the umbilical artery. So tiny. So these are the walls, and we can measure those diameters and get measurements of the velocity of the blood, so we can determine the flow, the blood flow in the placenta. So Catherine Dibben, a former student of mine, determined the impact of microplastic exposure on placental blood flow. And interestingly, she saw that there was an effect in both the microplastic and nanoplastic exposure group, but in the opposite direction. So in the microplastic exposed group, there was an increase in blood flow, and in the nanoplastic exposed group, there was a decrease. She also looked at blood flow to the brain. The reason that we do this, and this is done in humans as well, is if the resistance to blood flow in the brain decreases, this suggests that blood is being redistributed to the brain at the expense of other organs. This is a phenomenon that's called brain sparing, and it often occurs if the fetus is in distress. And it can be maladaptive. It's a good thing to do in very short situations, like if the umbilical cord was to kink, the oxygen would be redistributed to the brain to keep the brain safe. But in long term, it's shown to have negative effects on the fetus. And in both the microplastic and nanoplastic group, we saw this impact, that there was a decrease in resistance, suggesting that there was brain sparing occurring in these fetuses. All of this work was done on polystyrene as a plastic. That was just what was available that we could purchase and what everyone was using in the field. We wanted to see, is there an impact on other types of plastic? Polyethylene is one of the plastics that we make a lot of globally. So Jenna looked at whether there was an impact on polyethylene exposure. And she did not see an impact on the fetal weights for the polyethylene exposure group. That's what's shown here in yellow. There was an impact on placental blood flow, and there was not a significant impact on the resistance in the brain. So it does seem like the developmental toxicity does depend on the polymer type. This is for the non-chemists, I guess, looking at polyethylene versus polystyrene. There is some literature that suggests the aromatic group is increasing the toxicity of the polystyrene. This is obviously a space where a lot more work needs to be done, right? We have lots of different plastics to look at, and we're really trying to work towards looking at mixtures of plastics that would better represent what humans are exposed to. Okay, so my group with an interest in metabolism wanted to know if the fetal growth restriction that we're observing was partially the result of alterations in placental metabolism, and also was there an impact on the fetal brain? So placental metabolism is something that we know is important for proper growth. This is a great paper from a group out in Ottawa, just talking about in humans, the possibilities for placental metabolism to be used in pregnancy research for screening, diagnosis, understanding the etiology, lots of options. So our approach is to use the NMR facility, which is right across the hall. Because of my background in solid state NMR, we use magic angle spinning NMR. So we collect our samples, we instantly freeze them in liquid nitrogen, and then we really just take unprocessed, intact tissue, insert them into the rotor to try and avoid any extraction procedures, minimize the impact on solvent effects, and try to represent the molecular integrity that we would have in vivo. And then, yeah, we use our 600 megahertz machine. We spin the samples very slowly, well, slowly relative to more materials chemistry applications to try and maintain the tissue integrity. I won't go into too much NMR background, but basically the reason that we spin the samples is that if you have a solid and you don't do any spinning, then the dipolar interactions between the protons will just dominate the spectrum and you'll get this broad sort of hump. And so when we spin the samples, we narrow the resonances because we overcome that dipolar interaction. In tissue, of course, you've got a mixture of sort of solid and semi-solids and lots of water. So we don't really need to spin very fast to narrow our samples. So this is just an example of a sort of typical proton spectrum that we have from the mouse placenta. We were able to assign 14 different metabolites. We now have a brand new probe that my group has been working with that has allowed us to get even narrower resonances, and we're still working on our assignment. But this is what we've been working on for the past couple of years using just a standard solid state NMR probe. So this was again a project that Zara worked on. And this was on microplastics, and she found, as she increased the concentration of the exposure, that there was an impact on both lysine and glucose in the placenta. Both of these are really important for fetal development and for placental function. Glucose maybe is the most obvious one, right? It's the main source of energy for the placenta and for the fetus. So seeing this abnormal concentration at these high levels does suggest that there is an impact of the microplastic exposure on the placental metabolism. We do some multivariate analysis to try to, rather than looking at individual metabolites, how does it impact the entire metabolome? And we saw a sort of modest separation of the high concentration group compared to the controls. And then, of course, we can determine some of the pathways, the metabolic pathways that are altered with these exposures to give us a sense of a bit more information about what's happening. My master's student, or now PhD student, Grace, did some work looking at the fetal brain. So similar processing, but just for fetal brain tissue. And in this case, we've started looking at both females and males. And she saw an impact on multiple metabolites in the brain in both the males and the females. Glucose pops up again. These are all, again, important metabolites for brain function and neurotransmitters. So a negative impact. This was all just done at the higher concentration in this case. Okay, so I'm going to tell you about one more microplastic study design. This was a bit more complicated. So we start with we set up the mice to breed, then they receive either controlled drinking water or drinking water with the nanoplastics at the higher concentration. We wanted to study what happens postnatally because everything we've done up till this point was in utero. So the pups are born, we do some developmental milestone testing. So we look, is there any impact on what's called the writing reflex? So all of these developmental milestones have a very strict age where they come in, where the fetus, where the pups develop them. So they should learn to right themselves at a certain day. So we test when they were able to do this. We also looked at when their eyes opened, and then right at weaning, so equivalent to adolescence in humans, we collected their brain tissue to study them by MRI. So we didn't see any impact on the writing reflex, which would be an indicator of motor function, but we did see that there was an impact on when their eyes opened. And this was sex dependent. So in black is the control data, in red is the nanoplastic exposure, and in the females, there was no impact on the time that the pups opened their eyes, versus in the males, we saw that they opened their eyes earlier in the nanoplastic exposed group. At first, I was a bit surprised by that because usually in environmental exposures, we see a delay in terms of when they reach their developmental milestones. This has been reproduced in another paper that was recently published, not by our group, but by another group. And once I dug into this literature, there's a lot of examples where developmental milestones happen early and are also concerning in terms of the timing is really important. So early or late, not good. Okay, so then I told you we took the brains and we ran MRI on them. This work is done at SickKids in Toronto. We don't have an MRI preclinical scanner here yet, but I continue to advocate for this and hopefully we will see one in the next couple of years. I'll walk through what these images look like. So this is a sagittal slice through the mouse brain. This is the olfactory bulb here. Here's the cerebellum. And each of these slices corresponds to a coronal slice through the brain. So any of the color coding that is blue on top of the MRI image is areas that were smaller in the nanoplastic exposed group compared to the controls. And areas that are red are areas that were larger in the nanoplastic exposed group. So we saw decreases in the olfactory bulb, large decreases in the medulla, and there were increases in the striatum, areas of the motor cortex, some very small bilateral areas in the hippocampus. We also found that some of these changes were sex specific. So here, this rainbow color map is showing areas where there were differences based on whether they were female or male. For example, I show here the visual cortex. So there was not an impact on the females, comparing the control females to the nanoplastic exposed, while in the males, there was a significant decrease in the visual cortex. And I bring that up because that was interesting in light of the difference in the eye opening that we saw in the males, but not in the females. I don't want to say that there were only changes in the males because, of course, there were areas that were more impacted in the females than the males. For example, in the medulla, we see that large decrease was mostly driven by the females. Okay, so to summarize the work the work we've done on microplastics in humans, we've shown that the microplastics in the human placenta, there's a very large variation in concentration that hopefully the fields will work together to figure out why. In mice, we've shown that maternal exposure to microplastics and nanoplastics significantly impacts early life development. So we've seen growth restriction, abnormal placental metabolism, and abnormal brain development. The developmental toxicity appears to depend on the plastic type and size. Lots more work to be done there. And I will argue that it's very critical to better characterize exposure levels in both healthy and complicated pregnancies going forward. Okay, I'm going to tell you about the second class of environmental toxicants that my group is interested in. These are poly and perfluoral substances called PFAS. You may be very familiar with PFAS. They have this long carbon chain with fluorine molecules rather than hydrogens. And because of this carbon chain with the fluorines attached, they have very unique properties. So they're used in nonstick cookware, cosmetics, clothing, lots of different applications. PFAS are persistent, bioaccumulative, and toxic. And as a result of this, many of them have been, well, several of them have been banned by the Stockholm Convention because of links to health issues, including this particular compound here called PFOA. You may recognize this man. This is Eddie Shear, who was famous, has always been famous as our meteorologist. But very recently, he was in the news because he lives in Torbay, close to the airport, and his drinking water is contaminated with PFAS, PFOA and PFAS in particular. This is as a result of firefighting training that took place at the airport. It stopped, I think, in 2001. So here's the St. John's Airport, and Eddie lives in this community here. So over 20 years ago, this firefighting foam got into the ground, and it's traveled through the ground and through the water into his well water and his neighbors. And so we are working with Eddie to try and do testing of, right now the government is only testing this very small neighborhood, and we're very interested in testing different areas throughout Torbay to get a sense of how far the PFAS exposure how far it has traveled. So this is a project that we just got ethics approval in January, and we've started this work. PFAS is not just in Torbay, it's a topic that you'll see in the news quite often. They're referred to as forever chemicals because they don't degrade in the environment and they don't degrade inside of your body. It's predicted that there are over 12,000 PFAS of interest that are currently not regulated or have been understudied. This is a group of chemicals here called fluorotelomere ethoxylates that our group has recently detected in dust samples collected in St. John's and in industrial wastewater samples collected in Ontario. These compounds are not regulated, but you can see the similarities, right, between the chemicals that are regulated and the chemicals that are being used by industry and are now in the environment. So our question is really, what is the developmental toxicity of these fluorotelomere ethoxylates and chemicals like this? We've shown that they are persistent in the environment, so we want to understand their toxicity. So similar to the microplastic study design, here we set up our breeding in the mice. We have a control group. Here we looked at 2 concentrations of the legacy PFOA chemical that is regulated, that we know is toxic, and we compared it to similar concentrations of the fluorotelomere ethoxylates. We chose 5 nanograms per liter as our low concentration. That's the US EPA regulation limit. Canadian regulations are a bit different. We don't have individual limits, and we really only have guidelines at this point. This is something that we're, as a community, are trying to fight against. And then 100 nanograms per liter we chose because that was the level we found in the industrial wastewater. So that was our sort of high concentration. This was the work of my student, Katie Steeves. She looked at fetal weights, and at these low concentrations, we didn't see a significant impact. We did interestingly see an impact on placental weights. So in purple is the fluorotelomere ethoxylate group compared to the PFOA in green. And she saw an increase in placental weight, which corresponded to a decrease in the placental blood flow. Often, if the placenta isn't working very well, if it's inefficient, it will continue to grow to try and compensate for the fact that it's not delivering enough nutrients and oxygen to the fetus. So that's what we think is going on with this increase in placental weight. We did some metabolomic studies, and we saw the metabolism was impacted in both the PFOA and fluorotelomere ethoxylate group, and not always in the same direction. So we saw there was an impact on asparagine, for example, decrease in lysine, while in the fluorotelomere ethoxylate group, there wasn't any significant impact on glucose, but there was in the PFOA. So despite the similarities in structure, we do see differences in terms of their impact on development. This is just some of that multivariate analysis where here, based on the full placental metabolome, we were only able to see separation between the fluorotelomere ethoxylates and the control group. So all of that data together, and we're still doing a lot more work in this space, it suggests the fluorotelomere ethoxylates are having a negative impact, and if anything, might be more toxic than the banned chemical PFOA, at least from the measures that we've been looking at. So I would summarize that portion of the study to say that maternal exposure to PFAS we know causes developmental toxicity. The causal pathways and the mechanisms of toxicity seem to be different. This could be due to a number of different things, variabilities in terms of that chain length. We also, of course, have different N groups. We've been working with Myrna and Andre Simpson at U of T, who have done a lot of this type of work in Daphnia to try to understand these differences. I'll say that while meant to replace PFOA, these fluorotelomere ethoxylates may be causing even greater toxicity. And all of this is very important to study metabolism because we know that these early metabolic changes can impact later life. And so, of course, we need new environmental regulations and guidelines on these replacement PFAS. And the Canadian government is working towards this, but it's moving fairly slowly. Okay, so that is it for me today. There's lots of people to thank, including some of the people in the audience, and I'll thank the funders, and I'm happy to take questions.
All right, thank you so much, Dr. Cahill. Do we have any questions, either in person or online? If online, you can just type in the chat.
Yeah, very interesting talk. So the earlier slides about the differences between your study and New Mexico. So your feeling is that they're using very different methodology. So it's not the actual different signal that people in New Mexico are high on plastics as opposed to people here.
I mean, I imagine there are geographical differences in terms of exposures, though we don't know that yet. They're using the same mass spec technique. We've tried to reach out to this group, and other groups have tried as well to understand. They don't provide a lot of details in their methods. So these are samples they've collected over 10 years ago. So they're in plastic containers, for example. They don't report their blank concentration. Their validation of their method and their quality control is not usually described in their papers. And if you try to contact them, they're not great about sharing that information. So it's hard to compare when people have kind of put up barriers about. I imagine that some of that is background. I don't know. Hopefully there's no one here from.
Yeah, no, so but that would be a problem with getting them for the intercalibration, because intercalibration probably will mean one of the paper has to be retracted. So they may not be motivated for this.
I think their levels are overestimate, like they're an overestimation. Now, if we were doing everything the same in terms of the quality controls and the analysis, it may still prove that their levels are higher. But I have a lot of questions for them, and we're not getting there in terms of, we're not getting answers, I guess.
So would the storage over a longer period of time will contribute microplastics from thing, or those existing will differ, or? We
haven't tried that ourselves. Because we kind of joined at a stage where we started collecting our samples in glass, we haven't tested this. Our collaborators in Amsterdam have suggested that, yes, because you are sort of picking away at the vial, so little bits of plastic, I think, are going to contaminate. I don't think that's the only reason that we're seeing differences, but we actually haven't tested it in a systematic way, comparing a glass versus plastic sample. I think the biggest, there's a lot in the air, right? So even working in, I don't know, we don't get not to get into detail, but like the placenta has to come out and then we get it in a metal vial, but there can still be plastics falling on it as this is happening. So there's a lot of potential sources of exposure, which is why those blanks are so important, right? We have to subtract out what the placenta is getting exposed to all the way along.
So do they have blanks?
They say they have a blank and they say there's nothing in it, which is not our experience. So
yeah, so especially if you're expecting that environment has so much of the stuff there, then blank shouldn't be empty.
yeah, I agree. That's what I have had
opposite
I recently had the opportunity. So the brain paper is published in Nature. I've already got down this, but is published in Nature Medicine, right? Which is a very high impact journal. But I recently did have an opportunity to review and ask these questions of them to ask about blanks. And so we'll see what happens in response to the reviews. Be critical, I guess, students. Be critical of methods, right? Just because something's published in a good paper, a good journal, doesn't mean that it's.
Yeah. Thank you. Cool talk. I'm just curious, you said there's speculation as to why male fetuses and female fetuses respond differently. Can you talk a little bit about that?
Yeah, so one of the main theories that I think makes sense is, so male fetuses are usually bigger. They have a faster growth trajectory compared to females. So I've seen this in the mouse. If everything is normal in a healthy pregnancy and then we introduce an insult of some sort. So I've done hypoxic insults where I just decrease the level of oxygen that the mother's breathing. The female fetus adjusts very easily. It redistributes its blood flow and then it goes back to completely normal. Versus it's like the male is using all of its resources to keep growing, and then you introduce an insult and it goes into distress right away. So I think it's the female has more ability to compensate for stresses that the male doesn't. And like I said, we've seen that in the mouse. In the human, it's harder to control in that way. But that's one of the theories, which I think to me makes sense, that the male's just using more resources.
Okay, and we have a question online from Dr. Suzanne Dufour. Thanks for a great talk, Lindsay. I'm curious to hear more about possible explanations for the differences in male and female fetuses observed in your micro and nanoplastics studies. She also says that she highly recommends Holly Hogan's book as well.
Yes, fantastic. We actually were we had Holly Hogan come to a panel discussion that we did. And yeah, she's so passionate about this topic. But yes, Suzanne said, so I hope I just answered that question by answering Catherine's question about the differences.
Yes, sorry about that.
No, it's okay.
Is there any other questions in the audience or online?
Growth components in males may be more vulnerable because of the hormonal mimicry.
Of course,
So
yeah.
would it be, let's say, leaching out of the plastics into the components which may affect the male hormonal balance differently than female?
Yeah, the example of growth is just one example. I just put a CIHR grant in for this to investigate the impact of chromosomes versus hormones. So I don't know if people are familiar, there's a mouse line called the 4-core genotype where you can separate out chromosomes and hormone contributions. So you'll have an XY with testes and an XY with ovaries. And so you can study the impact and determine whether hormones are, I don't want to say to blame, but the hormonal impact.
Mechanisms for the input.
So plastics, there are quite a few people who are, exactly, that'll answer mechanistic questions. There is a lot of literature showing recently that microplastics are endocrine disruptors. So certainly I think hormones have a role to play in this. We don't know the answer yet, but I think it's important to study. So yeah, thank you for bringing that up.
All right, is there any other questions? If not, thank you very much, Dr. Lindsay, Dr. Cahill, for your talk today. I'd also like to remind everybody here that BGSS, Biology Graduate Student Symposium 2025, registrations close next week. So if you'd like to present, please apply. All right, and thank you. well.